Hi, we are PhD students who come from different backgrounds. Some of them are biologists, biochemists, some of them are biophysicists, and mathematicians. We come from different countries as well. My name is Bù Minh Truong. I'm from Vietnam, and I'm here to talk about the project that we are working on to the public. It is believed that scientists are some people who usually say something that are not easy to understand. But no, science is understandable and thorough. And we are going to work with the food poisoning caused by bacteria, and bacteria are our enemy. To fight against their enemy, we need some tactics and tools. We should know who are our enemies, how they look like, what are their weapons, what are their weaknesses. And my mission is to look for their weakness. And that is the protein complex of the bacteria that help them survive in a harsh environment. I will measure where are they and in the bacteria cell. I'm going to use the microscope. Imagine that the cell shape is a cycle, and I will see whether these proteins complex are in the edge or in the center of the circle, and how they move in the cell in the top circumstance. In other words, we are going to open the door of victory, but there are millions of doors in the maze. My work is to determine where is the right door lead to the enemy. I will light up the door and put the color marks on that so that my allies can open the door. Now move on to a bit detail with my friend Alcridas.
I am Algirdas. I come from Lithuania. I am currently based in Germany. I believe we should use quite sophisticated methods for, fighting food poisoning, because you can use a variety of things for, setting your food, making your food sterile. You can set it on fire or boil it to hell. But I believe there are more generous ways of processing your food. So we're trying to fight bacterial contamination in foods. And yes, my colleague Bù Minh helps localize particular targets inside cells, like proteins. And I take these out of the cell and I try to look at what they look like, because each protein is sort of like a special snowflake, and each one is unique. And I will use a method. It's quite a cool new thing. It has won the Nobel Prize this year. So it's called cryo-EM, cryo-electron microscopy. So we're going to take this protein out and we're going to freeze it very quickly so that, ice doesn't even form crystals. So it looks liquid, but it's still solid. And we're going to blast the hell out of it with a very strong electron beam. Because I'm half physicist now, so I like my microscopes a bit stronger and a bit bigger. So we have a 3-meter high microscope back in our lab. And with this, we'll get these 2D images, so it's kind of like shadows of all these proteins. And from these, if you look at a hand on different sides, you can see, is it a side or is it the 5 fingers that you see? You can kind of reconstruct how a hand looks, or how a protein looks for that matter, from its shadows. So that's what we're going to try and do, to get the general idea of what we have and what we're dealing up against. And I will get a very broad overview of this. And if we want to look into more details about this, we will have to resort to X-ray crystallography, which is Sema's duty here.
So hi, I am Sema and I'm coming from France and I'm doing my PhD at Newcastle University in England. So in this project I am the structural biologist. But what does a structural biologist does? So the structural biology try to find out protein structures to understand how they are working. So to do so, I'll use the method called X-ray crystallography. So the crystallography part will allow to have the protein in an ordered way. So to illustrate, we can imagine that you have building blocks and you build a house. So you can definitely see that it's a house. But if you spill your building blocks on the floor, you cannot tell if it's a house. Having this, we'll then use the X-ray part that will be used to blast the crystals with the X-rays, like the doctors does to see your bones in your bodies. And finally, discovering the protein structure will help us to understand its activity and help us to stop it from spreading in our body, in our food.